Okay, so yeah, good morning everyone, from the UK at least. Welcome back to WebEVTalk after a short break that we've had. And just to say, as usual, we've got for our European sessions, we've got our co-host Jan Lötvall today, and myself, Aaron Scott. I'll be hosting today, but don't worry, Carolina, as we can see, has not gone too far and is ready in the wings if we need her. Today is the 4th of February, 2021, and we have a really interesting topic and what I think will be a really stimulating talk from Ian McGough today. So Ian completed his PhD at the University of Bristol in the lab of Peter Cullen, where he investigated the role of the retromer trafficking complex in facilitating Wnt secretion. He subsequently joined the lab of Jean-Paul Vincent at the Francis Crick Institute to study how Wnt proteins, despite being lipidated and poorly soluble in the kind of aqueous extracellular space, how it's able to diffuse to form concentration gradients that are essential for tissue growth and patterning during development. So I guess I can say this postdoc post in many ways culminated in an exciting and quite provocative paper that was published last summer, which in part at least I think Ian will be covering in his talk today, with the title and question, extracellular vesicles in developmental signaling, are they necessary for diffusion of lipidated Wnt? So please welcome Ian. Please go ahead and share your screen, Ian.
Yeah, good morning everyone, or afternoon depending where you are, I suppose. Before I start, I always like to acknowledge both Yvonne Jones at the University of Oxford and her PhD student Luca, because this was very much a collaborative effort, and it was very, very much fun working with both of them, and they were both essential to the story. So right from the off, I always like to acknowledge them, and you'll be seeing some of Luca's work later on in the talk. But what you're seeing here in this background image is a field of Wnt signaling molecules that are being produced by these cells here and diffusing away. And what I really, really care about is how these Wnt molecules actually get from A to B, which may sound like a very, very trivial question, but you'll see in a minute why it's actually a very interesting question and why it's been studied for probably decades now. So in the Vincent lab, first, just to give you an overview of what we care about is organ development. And when you think about how during development you go from a couple of cells to a large organ that's patterned correctly, grows, reaches the right size, how does this actually happen? And one of the ways that's facilitated is via morphogen signaling. So I like to make this analogy of a building site. If you're on a building site, it's pretty much like building an organ. You need to communicate to the workers so they know exactly what they're doing and they know what instructions to follow. So these cells here are kind of like our foremen on the building site, sending out instructions to all of the workers so they know exactly what they need to do. And it's kind of like this in organ development. We have a set of cells or foremen that secrete signaling molecules. And these signaling molecules diffuse away and they tell the surrounding cells exactly what to do. And this way, the cells adopt the right fates and carry out the right functions during development. But how do morphogen signaling molecules like Wnt actually do this? Well, the way they do it is by forming a morphogen gradient. So here's our foreman secreting a Wnt protein in this instance. And the Wnt protein diffuses away and it forms a concentration gradient. And depending where the surrounding cells are sitting in this concentration gradient, they'll activate distinct transcriptional programs. So these guys here, for example, that are receiving lots of Wnt will activate a blue transcriptional program. Those receiving very little, a red transcriptional program. And in this way, Wnt proteins and morphogens in general can tell the surrounding cells exactly where they are and help drive tissue growth and cell fate specification. And an example of this at play is in the spinal cord patterning. And in this tissue, we actually have 3 morphogen gradients at play. So on one end, we have a gradient of BMP molecules and Wnt, forming a gradient in this direction. And coming from the opposite side, we have a gradient of sonic hedgehog. And the cells here sitting in these fields will read the concentration of the various inputs, and they'll activate distinct transcriptional programs and adopt the right cell fates. So for example, this guy down here that's receiving very little Wnt and BMP, but a lot of sonic hedgehog, another morphogen, he'll become a motor neuron. So basically by reading out these gradients, cells know exactly what they need to do. And in the Vincent lab, we use Drosophila to study organ development. And the organ of choice for us is the wing imaginal disc, which then becomes the wing of the adult fly. And this tissue starts off as about 30 cells in an embryo. And then once the embryo hatches, it goes through 3 larval stages before it pupates and then emerges as an adult. And during these larval stages, these 30 cells undergo a lot of cell division and tissue growth, going from 30 to 50,000 cells approximately. And it's a great system to study organ development because there's so many signaling pathways at play. So we've got Wnt, BMP, and hedgehog, the ones I mentioned in the previous slide. But we also have EGF, we've got systemic signals such as insulin, nuclear hormones. So it's a really powerful organ to study development. And I'll just show you 2 of the morphogens that we care about in the lab. So here's BMP, which imply the protein we care about belongs to the BMP family is DPP. And DPP is expressed here and spreads away, and it represses this its target gene brinker. And this allows the tissue to grow. But also this gradient also helps specify the veins on the wing imaginal disc. And the guy I'll be focusing on for the rest of the talk is wingless, which is the main Drosophila Wint. And it's expressed here at the dorsal ventral boundary of the wing imaginal disc. And it spreads away and activates its target genes such as vestigial. And this helps drive tissue growth, but also it helps drive specification of cell fate. So if you notice at the edge of the wing here, we've got a lot of bristles and hairs, and it's wingless that actually specifies this hair formation. And it does so by activating a target gene along the DV boundary called senseless, which is a very high target gene. So we can see that wingless actually has quite a long range here. It spreads over 15 to 20 cells, and it forms a concentration gradient activating its target genes. But how does it diffuse, and why is diffusion potentially an issue for Wnt? And the reason is that Wnt are actually lipid modified, and this has been known for almost 20 years now. And possibly the best example of it and the importance was when Wnt was crystallized, bound to its signaling receptor frizzled. And in this crystal structure, you could actually make out the lipid moiety, or density representing the lipid moiety. And the lipid was engaged in a CRD domain of the frizzle signaling receptor. So the lipid is actually crucial to Wnt's function, because without it, you can't engage the signaling receptor and activate signaling. But this lipid is very, very hydrophobic. So it raises the question of how do you actually diffuse a way to reach your signaling receptor? And it also kind of creates a second problem, because you must shield the lipid in order to diffuse, but the signaling receptor then needs to bind the lipid to activate transduction. So not only do you need to shield the lipid to diffuse away, but you also then somehow need to uncover it to activate signaling. And just over here, you can see my schematic, which I'll be using to represent Wnt during the rest of the talk, with a kinked squiggly line representing the lipid moiety. So how exactly does Wnt, which diffuses long distances, get from its sending cells, move through the aqueous space, reach receiving cells, and uncover the lipid again to activate signaling? And this has been the main focus of my postdoc in JP's lab. So just to remind you the setup, because it's going to be important for all the remaining slides going forward. This is our organ of interest, the wing imaginal disc. And in this organ, the source of Wnt we care about is expressed here at the DV boundary, and it diffuses away to form a gradient. So how is the lipid shielded to basically facilitate this diffusion? And this isn't a new question. It's been puzzling the field for a long time, and a lot of labs have studied it and put forward a lot of models to explain how the lipid could be shielded. And I'm just going to talk you through the various models that have been proposed and show you some of my own data on them. And I'll focus on exosomes and lipoprotein particles today, because these are both extracellular vesicles, and given the target audience, they're the ones that will most interest you. Well, I'll start off with this guy who I'll briefly mention, but I won't show any data. But one of the models that's been proposed is that Wnt are bound by this protein called secreted wingless interacting molecule, or SWIM. And these proteins have a proposed lipocalin domain, which binds lipids. And the idea was that SWIM proteins would bind the lipid moiety on Wnt, shield it, and then these two guys would diffuse away together in the extracellular space, with the hydrophobic lipid kept happy in its pocket in the SWIM protein. I won't go into it today, but one of the first things I did when I started in the lab is using CRISPR. I made a fly that's mutant for SWIM, and the fly is viable, happy, fertile, makes it to adulthood, and no obvious effects on Wnt signaling or Wnt diffusion. So I'll just rule him out now, but I won't go into it any greater detail. And all the other methods or models that have been proposed to facilitate secretion and diffusion involve what I'm going to call group travel. And this is basically Wnt traveling on higher ordered structures. And obviously the one that will probably be of most interest to you is exosomes. And I'm going to go through each of these now in greater detail in the next slides. And I'll start with exosomes. Given the people I'm talking to, I probably don't need to explain this because you guys probably know much more detail than I do. But just in case any non-exosome experts or Wnt people have snuck in, I'll give a brief overview of what exosomes are. So following endocytosis, endocytic vesicles fuse with each other and fuse with early endosomes. And this early endosome is then essentially like a sorting station from where you basically sort your receptors that you want to go to different destinations in the cell. So if you want certain receptors to go back to the plasma membrane, they'll enter recycling tubules and go back there. Or if you want to bring something back to the Golgi, it'll be sorted and go to the Golgi. But the sorting step that we care about for this talk is actually degradation. So what happens is that you get inward budding on these endosomes, where you sort predominantly ubiquitinaceous cargo into these intraluminal vesicles. And normally what happens is these endosomes with these proteins targeted for degradation will fuse with the lysosome. And this will empty the intraluminal vesicles into the lysosome, where they'll be degraded and you can recycle the amino acids and so on from the lysosome. But occasionally these endosomes with these intraluminal vesicles will fuse with the plasma membrane instead of the lysosome. And this will release the intraluminal vesicles into the extracellular space. And then they're termed, in essence, exosomes. And you may notice up here, we have what micro vesicles instead of exosomes. And I'm briefly going to mention these because these have actually been reported to be the main method of diffusion for another morphogen, hedgehog, which is also lipidated similar to Wnt. And basically the difference between microvesicles and exosomes is that these bud from the plasma membrane, almost like a virus, and they tend to be bigger. But a lot of the same machinery is needed for the formation of both. So escort proteins, for example, have been linked to both exosome formation and microvesicles. But if we go back to these exosomes for a minute, once they're secreted, they're known to carry a range of different proteins and molecules, DNA, RNA, some of the escort components that are involved in their formation. But of interest to us is actually signaling molecules. So either within the exosome, or in this case, as has been proposed, you would have Wnt on the surface of the exosome, with its lipid maybe embedded in the membrane or bound to another signaling receptor on the surface. So this is a very attractive model for Wnt diffusion because Wnt's are known to be endocytosed and transcytosed through the tissue in wing imaginal discs. So they will be passing through the endocytic network, and there will be the perfect opportunity to load the Wnt onto intraluminal vesicles. And with the lipids embedded in the membrane or bound to another receptor, the lipid would be kept happy and Wnt could diffuse away, content and soluble. And when I started in the lab, another postdoc, Karen, had actually been working on this problem for a while. And she wasn't the only one. There's lots of groups in the Wnt and hedgehog field that had been looking at whether these molecules are found in exosomes and whether exosomes are required for their diffusion. And one of the first things Karen did is take a cell line stably expressing the main Drosophila Wint wingless. And as a control, she has this cell line not expressing Wint. And Karen basically took the medium from these cells and did a couple of low-speed spins to clear debris, and then did a 100,000 G spin to basically pellet the exosomal fraction, stain for Wint, and look if Wint actually finds its way onto exosomes. And she could actually recover a decent amount of Wint in the P100 fraction. And she actually quantified this, and it's not a tremendous amount. It's probably about 10% of the Wint compared to what's left behind in the supernatant after pelleting the exosomal fraction. But if Karen took this fraction and ran it on a sucrose density gradient and isolate the fraction that corresponds to exosomes, and then use this fraction to EM, she could see exosomes via the EM, and she could also see Wint present on the surface of these exosomes. So it looks like a pool of Wint does make its way onto exosomes where it's bound on the surface. But then Karen was quite disappointed, because she then actually said, okay, is this pool actually signaling competent? And then it got a little bit deflating for Karen. So again, here's our control of the S2 cells not expressing Wint. And here she's purified the various fractions. So this is the S10 supernatant that's just left behind after clearing debris, the supernatant left after pelleting the exosomal fraction, and the pelleted exosomal fraction. And you can see after purifying these exosomes and adding them to a Wnt luciferase assay, there was very, very low signaling activity, barely above the background from cells not expressing Wnt. And most of the signaling activity was still present in the supernatants left behind. But if you
just a very quick question.
Yeah.
How long was the spin?
This is Karen's work, so I'd have to go back and look. Off the top of my head, I have no idea. But it's if you are interested, you can check it up in her paper. But it's a good few years old now, so I can't remember off the top of my head this spin length. But if Karen concentrated the P100 fraction and really forced the issue with lots of purified exosomes, she could actually get signaling activity, showing that this Wnt on these exosomes could signal, although poorly. So there's a lot of caveats with this approach, is that such as these S2 cells actually don't normally secrete Wnt. They're being forced to express Wnt, so maybe things behave differently. So the obvious step was to go in vivo and actually look, do we need exosomes and can we see Wnt on exosomes in vivo? And again, you guys will know this better than I do, but just to give an overview of the exosomal formation, because I'll be targeting some of these in vivo. Here's our escort complex, and there's various different subcomplexes of escorts, and I won't go into the details, but escort basically recognizes ubiquitinated cargo that's targeted for the degradation or target for exosomes, and it basically clusters this ubiquitin so that it can be incorporated into this forming interluminal vesicle. And the other protein I'm going to point out here is VPS4, which is important for constricting the neck of these interluminal vesicles and facilitating the scission process so they can form. So we decided to target some of these genetically and look what happens to Wnt secretion in the gradient. So the first one we targeted is HRS, a component of this escort 0 complex. And just to familiarize yourselves with how we do these experiments, it's very neat. In Drosophila, we can always have an internal control. So what we do is we use transcriptional activators that are expressed specifically in only half of the wing organ. So all our genetic manipulations are done here, and we have an internal control on the left-hand side. So we decided to make the posterior compartment mutant for HRS. And when we did this, the mutant cells are marked by the absence of GFP here. And you look at the extracellular gradient of Wnt, you can see that Wnt is still getting secreted fine, still spreading away, forming a gradient. but there's a lot of redundancy potentially in this formation of exosomes. So we targeted a second protein, and this is VPS4. We overexpressed a dominant negative VPS4. And just to show you that the actual tool works very, very well, I stained for ubiquitin because it's predominantly ubiquitinated cargo being targeted for here. So if you interfere with this process, you'll accumulate ubiquitinated cargo. And this is exactly what happens. You get a massive accumulation of ubiquitinated cargo when you overexpress the VPS4 dominant negative. And again, we have our nice internal control here. And now if I come along and I look at what happens to the Wnt gradient upon this manipulation, you can see that the Wnt gradient is absolutely fine. It still spreads away perfectly okay. And I'll say Karen pretty much threw the kitchen sink at this, targeted various other components linked to ILV formation, RAB11, components escort 1 and 2, and we could never see any effect on the Wnt gradient. So this kind of, for us, was saying exosomes aren't involved. So we turned to the second carrier, extravesicular carrier that's been linked to Wnts, and hedgehog as well, actually, diffusion. And this is lipophorin lipoprotein particles. And basically these lipoprotein particles are a way of transporting lipids around an organism, because lipids are hydrophobic, just like the lipid on Wint, and you basically need to shield them so they can diffuse and reach various organs in the organism. And what happens is you get the really hydrophobic lipids such as cholesterol and tags in a hydrophobic core. And then you have this outer shell of phospholipids with the hydrophilic bit exposed on the outside and the hydrophobic bit in the middle. And this basically keeps the whole thing soluble. And then we have these apoproteins that kind of provide structural rigidity to the vesicle. And in Drosophila, all of these lipoprotein particles come from what we call the fat body, which is basically the fly's combination of the fly's adipose and liver tissue in one. And this fat body releases these lipoprotein particles into the Drosophila's blood system, the hemolymph. And it's been proposed that they would then reach the wing imaginal disc and other organs. And here they would be recognized by receptors and taken up. And it had been proposed that once these have been internalized, they would meet Wnt in endocytic structures. Wnt would be loaded onto these lipoform particles, which could then be resecreted with the Wnt happily soluble and content, because it would be embedded in the hydrophobic portion of these carriers. So we decided to genetically target these as well to see what effect they had on Wnt diffusion. And again, just to remind you, everything that's the manipulation is only in the posterior compartment, so we have a nice internal control. And what I've done here is I've overexpressed one of the receptors for lipophorin particles. And when you do this, you can see that you get this nice accumulation and uptake of the lipophorin particles in the posterior compartment. And the idea was, well, you're getting a greater uptake now, you're maybe depleting lipophorin particles from the extracellular space, maybe you're affecting the Wnt gradient. However, somewhat disappointing at the time, again, in the posterior compartment where we have this manipulation, Wnt was diffusing perfectly fine. And I'll say, as well as overexpressing these receptors, we also depleted the two main receptors, and we could also see no effect on the Wnt gradient. So at this point, we're quite disappointed, and we're scratching our heads and trying to figure out, well, how is it diffusing? And the last model that we had, the last main model that had been proposed, was that Wnt could be forming a kind of vesicle of itself, an aggregate or a meso. And basically, it would be forming a structure with the lipids facing in the way, kind of like a lipoprotein particle, a hydrophobic core, and the hydrophilic bit of Wnt on the outside. And basically, Wnt would diffuse away as a ball with the hydrophobic lipid content in the middle. And this is very, very difficult to test genetically, because it only requires the Wnts. So if you delete the Wnts, you have no assay. So I scratched my head for a while and thought of a way to test this. And we're lucky in Drosophila that we've got two endogenously tagged alleles of wingless, the main Drosophila Wnt. And these, I say we're lucky because these tags are actually, they don't perturb the function of the Wnt. and you get homozygous viable flies. And traditionally, it's been quite hard to tag Wnt and often kills its function. But in Drosophila, both this GFP wingless and this HA wingless are functional and behave like normal. So I got thinking, okay, we've got these two different tagged forms of wingless. If they're traveling together on these micelles or even on exosomes or lipoprotein particles, there'll be multiple copies of these Wints and these structures. And now if somehow I could stop one of these forms from diffusing, I could see if it affected the diffusion of the second form. And late in the last 10 years, there's been great advances in making nanobodies. These are small single chain antibodies that recognize various epitopes. And there's a very, very good one for recognizing GFP. And because these are genetically encodable, it means we can express them in vivo. And I thought, well, okay, if I make use of a known tool in the fly community called MorphoTrap, which basically is a GFP nanobody fused to a transmembrane protein, I'll be basically expressing a wall of GFP nanobody on the surface of the cells. And this would be basically just sitting there waiting to capture my GFP Wnt when it's diffusing past. And if I did this, I could then see, okay, I've captured the GFP Wnt. What happens to the HA Wnt? If they're diffusing together as a micelle, for example, when I capture the GFP Wnt, I'll also capture the HA Wnt. So I basically decided to give this a go. And again, just to remind you, the experiment is done in the posterior compartment. We have a nice internal control. And I decided to express this GFP nanobody everywhere in the posterior compartment, and then see what happened to the HA. And it works very, very nicely. So here you can see our GFP wingless and our HA wingless both diffusing away, forming a gradient in the wall type compartment. But in the compartment where I've expressed this nanobody, you can see that I've trapped and stopped the diffusion of the GFP form of Wnt. And although I've stopped GFP Wnt from diffusing, you can see the HA Wnt diffuses away perfectly fine, forming a gradient, no problem. And this suggests that Wnt is actually moving as a monomer. It's not forming micelles, or it's not being, multiple copies aren't being loaded onto exosomes or lipoprotein particles. And just to take this a step further and really put this tool to the most use, I decided, well, okay, rather than expressing it everywhere in the posterior compartment, I'll just express it in a stripe here through the middle of the wing disc. And I would capture the extracellular GFP wingless. And if I was capturing the wingless, I'd be also capturing whatever it's moving on. So here you can see nice capture of the extracellular wingless by the nanobody. If I come along and I stain for markers of lipoprotein particles, ApoL, you can see that I don't co-capture lipophorin particles. The same is true for exosomes. Again, nice capture of extracellular wingless. If I stain for HRS, a known exosomal marker, I see no capture of exosomes. And just to basically convince you that this tool actually works and that you can co-capture something, I expressed a secreted form of a protein called DLP, which is a glypican. And I'll tell you about glypicans in more detail in the next slide. But for this purpose, all you need to know is that glypicans are known to bind to Wnt molecules via sugar chains that are attached to the core domain of the glypican protein. Normally, these proteins are embedded in the membrane via GPI anchor. So I decided to delete this anchor. So now I have a diffusible form of this DLP glypican that's known to bind to Wnt. And when I express this V5 tagged DLP, when I co-capture my extracellular wingless, you can see that I co-capture and see lots of colocalization with this DLP molecule, which is a known Wnt interactor. So this tool works very, very nicely, and I can co-capture interactors, but I can't co-capture any sort of carrier or a second tagged form of Wnt. So at this time, I got very, very interested in this, this family of glypican proteins. And I'll just give you a bit of background as to what they are. And they've been known since the early 90s to have a role in morphogen signaling. And Drosophila have two glypican proteins, DLP glypicans and DALI glypicans. And as I said in the last slide, the most important or to this point, what was thought to be the most important feature of these proteins in morphogen signaling was the fact that they've got these sugar chains, these GAGs, chains attached to the core domain of the protein. And these are very, very charged sugar chains. And the idea, or what was believed to happen, is that Wnt, which is also very charged, would bind to these GAGs, and these Glypicans would basically retain signaling molecules in the ECM and stop their leakage or diffusion out of the tissue. And as well as doing that, these sugar chains are also known to bind to many of the signaling receptors that the morphogens bind to. So these Glypicans would, in essence, act as facilitators of ligand receptor interactions. And they have a very, very, so this is what I was saying, the Wnt was thought to bind to the sugar chains. And they do give a very, very strong phenotype on extracellular wingless. So in this experiment, I've made patches of mutant tissue that are missing both these DLP and DALI Glypicans. And you can see these mutant tissues, which are marked by the absence of GFP, that you see a dramatic loss in the extracellular wingless gradient wherever you're missing these Glypicans. But this doesn't solve our problem of the lipid moiety, because this is meant to be sugar-sugar interactions. So everyone kind of forgot about Glypicans in essence, and whether they could be involved in shielding the lipid, because obviously it's the sugars. But when I delved into the literature a bit more and actually started doing some experiments myself, I realized things are a little bit more complicated than this. Because if they're both behaving like this, they should be functionally redundant and interchangeable. But it turns out they're not. So if you make single mutant clones of DALI and DLP, here's a big mutant clone for DALI. You can see there's not really a dramatic effect on the extracellular gradient. You might get a small reduction, but nothing huge. But if you come along and make a DLP mutant patch of tissue, you can see a dramatic decrease in the extracellular gradient, suggesting that the two proteins aren't simply functionally redundant. And what makes this even more impressive, and I won't show you, is that DALI is actually very, very highly expressed here at the DV boundary, where DLP is actually very lowly expressed. So the fact DLP is giving this much, much stronger phenotype is basically astounding, given the various expression levels and patterns. And it's not just the loss of function where they differ. The most striking difference is actually on overexpression phenotypes. Again, if in my Drosophila wing disc here, my manipulation is just done in a stripe through the middle of the tissue. If you overexpress the DLP glypican, you can see that it leads to this massive accumulation and extension of the range of wingless, which is normally just here at the DV boundary. But if you overexpress DALI glypican, you don't get this, again, suggesting they're not simply functionally redundant. And the eureka moment, and I can't even take credit for it, it's been sitting in the literature for a long time, is if you overexpress the core domain of the DLP glypican, so missing the sugar chains, you can still get this capture, retention, and increase in the range of diffusion of Wnt, suggesting that the core domain can actually be involved in this process. I'm not saying the sugar chains aren't important, because it does dramatically reduce the ability to extend the range of wingless, but the core domain alone can do it as well. So at this point I got thinking, well, maybe it's actually like this. Yes, the sugar chains are binding to the Wnt, but the DLP core domain might have a lipid binding pocket. And this would be a way of shielding the lipid moiety of Wnt and keeping it happy in the extracellular space. So to test this, I've already shown you this setup of basically expressing DLP in a stripe through the middle of the wing disc. And what I did is I decided to use a GFP tagged form of Wnt. When you do this, it behaves like the endogenous. You capture it and you extend the range. But then I decided, okay, I'll mutate the lipidation site, so the lipid's no longer appended to Wnt, and see what happens to this unlipidated Wnt when you overexpress DLP. So here's the setup. When I do it, you can see that you do not see any capture of extracellular wingless when you're missing the lipid. And the reason I used the GFP tagged form of wingless for this experiment was it allowed me to do a nice control to prove that this protein gets secreted and is trappable, because I decided to turn to my friends the nanobody again and overexpress the morphotrap instead of DLP. And when I do this, you can see that, yes, you can capture this unlipidated form of wingless. You just can't capture it with DLP. So this is strongly suggesting that the interaction is lipid dependent. So at this point, we wanted some biophysical proof that this is actually the case. So this is where our good friends Yvonne and Luca come in, and some beautiful work that Luca did. So Luca decided to test this in greater detail, and he decided to do this using SPR, surface plasmon resonance. So what we did is we, through our fantastic collaborators in the Crick building, the peptide chemistry facility, we got small peptides made that correspond to the lipidation site of Wint, and we got these peptides biotinylated, so they could be used to coat a chip. And we either got the peptides made with the lipid moiety palmitoleate that's found on Wint, or without the lipid moiety. And then what Luca did is he purified DLP, and he purified the core domain of DLP, so missing the sugars now. And he decided to flow this across these chips and see if he could see an interaction. So if the DLP was binding, it would basically change the conductance on the chip, and you could get a readout of binding. And when Luca did this with the lipidated peptide, he saw a nice interaction, concentration dependent. So the more DLP you flew across, the stronger the binding. And the really positive result was the unlipidated peptide. When you did the same experiment, you saw no binding. So this is showing that DLP is really binding to the lipid moiety found on Wnt. And as a nice control, Luca took the core domain of DALI, which we've shown doesn't extend the range of wingless in vivo. And when he uses the core domain of DALI and he flows it across the lipidated chip, he sees no binding. So this is proving that DLP actually has lipid binding capabilities, and it's unique to the DLP family. So at this point, we're like, okay, is DLP the protein that's responsible for solubilizing and keeping Wnt in solution in the aqueous environment? So to test this, I turned to tissue culture, and it's been known for a long time that Wnt are very, very poorly soluble, and it's actually heartbreaking sometimes to work with them and actually get enough Wnt in solution. And you can see this here because I'm expressing a GFP wingless in these cells, and you can see there's lots of wingless expressed in these cells. But if you take the medium, and I'll say this is serum-free medium because serum is known to have some molecules that stabilize Wnt in solution. And funnily enough, those molecules are heparan sulfate proteoglycans like DLP and DALI. So that had been known for a long time, but no one thought it was lipid mediated. But in these serum-free conditions, you can see you get little or no Wnt in solution. Now, if I come along and I express my two Drosophila glypicans, missing their GPI anchor so I can get everything into solution and it's not just stuck to the surface of the cells. When I overexpress DALI, I'd see no increase in Wnt solubility, but when you overexpress DLP, you get this massive solubilization of Wnt in the medium. So this is really proving that DLP can be a solubilization factor for Wnt signaling molecules. And the really, really important thing is that if you take these fractions and you do a luciferase assay to read out signaling activity, you can see that Wnt purified from serum-free medium, you don't get any Wnt, there's no signaling activity. You add DALI, the same. The minute you add DLP and you take this fraction, you get massive activation of the signaling. So this actually importantly shows that DLP can bind and shield a lipid, but it can also release it again to activate signaling. And this gets to the heart of the second problem I mentioned in the introduction, that the Frizzle receptor needs to bind the lipid to activate signaling. So this is showing that DLP, yes, it can shield a lipid, but also it can let it go again so that the Wnt can bind to Frizzles and activate signaling. And this isn't just some quirk of Drosophila. If I take the vertebrate Glypicans, of which there's 6, 4 belonging to the DLP family and 2 belonging to the DALI family, and I do the same thing, I overexpress them in tissue culture. The DLP family members 4 and 6 lead to a massive stabilization of Wnt in solution, where the DALI family members 3 and 5 don't. So this is a conserved phenomenon. And just to prove this, I've shown you that DALI can't extend the range of Wnt, but DLP can. So if I express the vertebrate glypicans 3 and 5, which are the DALI members, just like DALI, they can't extend the range of wingless, but 4 and 6, the DLP family members can. And because high levels of these DLP glypicans bind the lipid moiety, they dampen down signaling because they're extending the range. And this actually inhibits high target genes, such as senseless, which I mentioned in the introduction, which is expressed just here at the really high levels of Wnt. And it's known if you overexpress DLP, you basically dampen down the signaling and you kill this senseless activation. If you overexpress DALI, you don't. And the same happens with the vertebrate glypicans. So I'm expressing them here again in a stripe through the middle of my tissue. When you overexpress 3 and 5, you don't affect senseless signaling. But because the DLP glypicans bind the lipid, they outcompete frizzled, you basically kill high signaling. And you can see that where they're expressed, they interrupt this senseless expression. But because they're sending the signal further away and aiding its diffusion, you actually end up increasing low target gene expression away from the source. So here I'm expressing the glypicans in the entire posterior compartment, and I'm looking at distal, a low target gene, and I'm comparing the posterior to the anterior. The DALI glypicans, if you're comparing where they're overexpressed to where they're not overexpressed, it doesn't really extend the range of signaling at all. But if you overexpress the vertebrate glypicans, or DLP, you can see that you get higher signaling further away from the source, and you get this shoulder on these plots when you're comparing signaling at a distance between DLP or DLP glypican overexpression from the vertebrates. So this is really showing that DLP and its vertebrate family members can bind, solubilize Wnt, and aid its transport. So now we wanted to go, okay, we know it binds the lipid moiety. Where does it bind it? So this is where Luca came in again. And Luca decided he was going to crystallize DLP, which had been crystallized previously. But this time he was going to add in our lipidated peptides and actually see if he could co-crystal the lipidated peptide with the DLP protein. And if you were familiar with glypicans before this, you probably would have put your money on it binding down here in this cysteine-rich domain, because these CRD domains on frizzled are known to bind the lipid moiety, but it turns out it's not actually. We have this lipid binding pocket up here in the C lobe. And if you compare Luca's structure to the previous structure, you can see that in the unlipidated, or the structure crystallized without the lipid present, you have these two alpha helixes right next to each other. But in the presence of the lipidated peptide, these two helices, alpha 9 and 10, they shift apart and they open up to allow access to this hydrophobic pocket. And if you look at this pocket in greater detail, you can see it's lined with very hydrophobic residues. So it's a perfect place for this hydrophobic lipid to embed, basically, and be kept happy. So then we wanted to really prove this is the lipid binding pocket and this is responsible for the solubilization. So we decided to make some point mutations to interfere with the pocket. I'm just going to focus on one today, and that's this double cysteine introduction. And the idea was by introducing a cysteine on this helix and this helix, they're close enough in the absence of the lipid to basically form a disulfide bond. And this basically clamps the two helices shut and it stops them from opening up to allow access to the hydrophobic pocket. And Luca basically made these mutants. He incubated DLP and the mutant with the fluorescent lipid, and he did size exclusion chromatography to recover his fraction of DLP. And the idea is if DLP is binding this fluorescent lipid, that fraction will be fluorescent. And then he could read out if these mutants were interfering with lipid binding. And this assay works very nicely. If you take wild type DLP, you recover lots of fluorescent palmitic acid in the DLP fraction. And if you look at this double cysteine mutant that we decided to introduce, it completely blocks the ability of this fluorescent lipid to bind DLP, as does another mutant that I won't go into today. So I decided, okay, I'll test this mutant in my assays. So I took the cysteine clamp mutant, went and looked at the solubilization activity of this DLP. So here you can see Wnt by itself, you get very little in solution. You add DLP, it's stabilized because of the lipid binding, lots of soluble Wnt. You add this DLP where you can't access the hydrophobic pocket, it no longer solubilizes and keeps the Wint in solution. And if you do the in vivo assay, again, just remind you, you overexpress DLP, you get this trapping and this extension of the range of wingless. And this is basically kills when you can't access the lipid binding pocket. So this is really proving that, look, DLP has a lipid binding pocket and it is responsible for solubilizing Wint and keeping it happy so it can diffuse away and form a concentration gradient. So just to summarize, this is how we see the whole process working. DLP will bind to Wint molecules via sugar chains in a charge-charge interaction, but it also possesses a lipid binding pocket to keep it happy and content in extracellular space. You'll have on-off diffusion, as shown by my luciferase assay, where this can be reversed, so it combines the receptor. And this Wint will then diffuse to the next DLP and so long, and move along basically in kind of in a bucket brigade fashion until it encounters a frizzle receptor, or frizzle levels are really high, and then it will activate signaling. Where DALI family members only have this sugar chain mediated interaction, so they can only act as co-receptors to present the ligand. And this also solves the problem of not just solubility, but also handover, how you free up the lipid to bind frizzled. And just an interesting thing, as I mentioned earlier, hedgehog is also duly lipidated, and DLP is known to have a role in hedgehog signaling. So I'm actively pursuing at the minute to see what role DLP plays in hedgehog signaling and whether it's also mediated by its lipid binding pocket. So I'd just like to thank everyone at the Crick, which is this shiny newish building in central London. I'd especially like to thank JP, my supervisor. He's been very supportive. Again, I'd like to flag up Yvonne Jones and Luca, who've been fantastic to collaborate with, and the peptide chemistry facility at the Crick, and especially Nicola O'Reilly, who without these peptides, we wouldn't have been able to do so many cool experiments. And thank you for inviting me and listening. And I'm happy to take any questions.
Thank you very much, Ian. That was really interesting. I'm sure there are, Aaron has a couple of questions, but I'll let Shiv in first. Shiv B, about the pockets. Please unmute yourself and ask a question.
Hello, thank you so much for the wonderful talk. I wanted to know, like, in the case of DALI, do you have the same hydrophobic pockets by any chance? And is that the reason, the absence of it, the reason for the lack of spreading of the ligand?
It's a really, really good question. And I'll actually just show a slide. It'll be the easiest way to answer it.
Sure.
So that would have been a good hypothesis, but it's actually not the case. So if you look at the pocket of DALI and its family members, it's also there, and it's also very, very hydrophobic. So it can actually accommodate a lipid moiety in theory. And there was some interesting residues that were different between DALI and DLP. So there was a couple of substitutions. So this F to T, F to Y, that made it slightly less hydrophobic. And we thought, okay, maybe the slight less hydrophobicity is playing a role. And there was also these putative glycosylation sites that, you know, if you add a glycosylation there, maybe you block access to the pocket. And I introduced these mutants into DLP to see if I could kill the activity. And when I did it, despite changing to these less hydrophobic residues or introducing this glycosylation site, DLP could still extend the range of wingless and still bind. And if I did the inverse and tried to make DALI like DLP, it didn't make DALI like DLP. So it's access to the pocket rather than the pocket itself. There's some intrinsic ability of DLP to shift these two helices open that DALI doesn't possess. But trying to map down the key residue, it could be somewhere else in the protein away from the pocket. It would be a nightmare to do. But it's a good question. The pocket is there. It just can't be accessed in DALI glypicans.
Aaron, you're up.
Nice one. So I'm sorry to sort of move the focus away from the work on the Glypicans. And obviously I thought that side of your work was clearly brilliant and incredibly thorough, as best as I'm able to tell. And I guess with my biases, and you're probably expecting this, overall, I'm kind of wondering whether EVs are worth revisiting in any way. So my thinking kind of being that the mechanism of biogenesis that you used to approach your knockout experiments were potentially sort of exosome specific, although I don't think there's any guarantee of that either. But whilst the isolation methods that found Wint in the EVs, from my understanding, would predominantly be made up of microvesicles. And I'm just kind of wondering then whether that could be the reason for the negative result in vivo. Yeah.
So microvesicles have been, there was a Nature paper a few years ago from the Thron lab, and they showed that in their case, hedgehog is targeted to microvesicles, but these microvesicles also depended on the escort components for formation. And if he targeted the same components, HRS or CHIMP4, another escort component, or VPS4, he saw an effect on microvesicle formation and hedgehog diffusion. So I think my manipulations, at least on the fly, and going by that paper, would be affecting microvesicle formation as well as exosomes.
And so kind of as a bit of a follow-up as well, I was just kind of, in my eyes, you can sort of see that the knockout experiments to sort of test all of these proposed mechanisms is not in any way straightforward. And I think I remember you mentioning in the paper that, for example, there's no way of preventing cytonemes passing Wint. So kind of thinking that with these difficulties aside, I was just wondering if you could sort of speculate on why these other mechanisms kind of exist. Why are EVs found to be containing Wints? And I was almost wondering whether could there be a degree of tissue specificity that these mechanisms might be needed in other instances, or
so first I'll address the cytonemes, and I didn't mention cytonemes because cytonemes are very difficult to test genetically, probably even more so than EVs, because the way to do it is perturb the actin cytoskeleton, and you'll have a lot of pleiotropic effects. But they have been linked to all morphogens and their diffusion, and I'm not against them, and they could be playing a role. And in fact, it would be complementary with the Glypicans, because the Glypicans could be sitting on these cytonemes, keeping the Wnt happy. It shoots out, and then that cytoneme transfers that Glypican at a distance, and it releases Wnt at a distance. And the Glypicans would be good carriers because of this GPI anchor, they're embedded only in the outer leaflet of the membrane, so they're quite motile and diffusible, and they can move rapidly in the plane of the cell. Cytonemes could have a role. I'm not going to say there is no role for exosomes or in other tissues. In Drosophila, for example, at the neuromuscular junction, which isn't an epithelium, it's been reported that exosomes transfer across the neuromuscular junction. And I've never looked there myself, but it has been reported. And I know in vertebrate systems, in the intestine, for example, Wnt has been shown to be transferred from the stromal cells at the base of the intestinal crypt to the actual epithelium, and exosomes have been linked there. So exosomes could be involved in other tissues. And to be honest, Glypicans could also play a role there as well, because GPI anchors are known targeting a motif for exosomes as well. So there could be certain tissues or in other organisms where Glypicans would target Wnt to these exosomes, and it would be the intermediate keeping the Wnt happy. I've seen no evidence in any tissues I've looked at for that, but I can't rule out that in vertebrates in the intestine, for example, that Glypicans are exosomal targeting motifs with the GPI anchor and aids that way. So I'm very open-minded.
Can I follow up on that question? If you have more questions, Aaron, I'll let you come back.
Yeah.
But it doesn't surprise me that the free molecule diffuses better than an exosome or an extracellular vesicles through the tissue, which contains other components and proteins and so on. So, to be honest, I'm not sure of the diffusion capacity of extracellular vesicles in different types of tissue. Have you tested that or thought about that?
So I haven't tested it because once the Glypican discovery rose, I put all my focus on that. but people have looked at it, and there are papers suggesting that exosomes might work in tandem with cytonemes for hedgehog signaling, at least. And people have reported seeing exosomes moving along cytoneme projections. They don't know whether this is moving along the surface of the cytoneme or whether it's inside the cytoneme, and the exosomes, basically endosomes, are transported up the cytoneme, and then exosomes are released from the tip.
Right.
So, to the best of my knowledge, no one's looked exactly at how diffusible exosomes are in the tissue. I do know in Wint diffusion, at least, most of the Wint has been shown to diffuse poorly, and you have a small fraction of maybe 9–10% that's quite soluble. And for me, that will be the bit that's currently unbound to the glypican, where the slower non-diffusing bit's the bit that's currently bound to the glypican. But yeah.
We have 2 questions in the chat box. Aaron, did you want to ask the last one, or have more?
Yeah, if that's okay, it's just more to a bit of to see if my interpretation is kind of correct from what you've presented. So I was just kind of wondering, with the DLP and the DALI mutations showing that they're clearly disrupting Wnt signaling, which I'm wondering, does that suggest that the other proposed mechanisms aren't even in that context, are not even sufficient to compensate when you sort of knock that down?
So I haven't shown, so I'm going to be cautious here, because most of what I've shown with the mutant is overexpressed, and what I've actually done now is I've made an endogenous DLP missing, or with the helices clamped together, and I haven't got around to checking that yet. I've just got it, and for me that would be the killer experiment. I would now have endogenous DLP that can't accommodate the lipid and see what happens to the range of diffusion. I personally, I don't think the other methods can compensate, and maybe this is, I'm going to turn this back, and this is a question for you guys, is one problem I've always had with the exosomes is this gradient turns over quite quickly. So if you do FRAP experiments, it's 1 or 2 hours. So you get a lot of Wnt being produced. I've always been puzzled, would it even have been energetically favorable for this small stripe of cells over the course of 2 or 3 days to be throwing away such membrane, such large quantities of membrane, and just energetically whether that would even be feasible. That I don't know. Maybe you guys could answer that one. Yeah,
I've definitely just a similar sort of veins of thought that I'm having about my own experiments and situations.
Yeah, how much exosomes do you actually find in the wing, developing wing?
So even the papers that report exosomes having a role, it doesn't seem to be, put it this way, they don't form the same gradient as wingless does. They might be a small pool. Yeah.
I'm actually not surprised. We have 2 questions in the chat, and we'll take those, and then we can come back to more face-to-face interaction. So Genevieve, please unmute yourself and ask your question.
Yeah, actually, you probably already partly answered it. I was just wanting to clarify. So the Glypicans are bound by their GPI anchor on the outside of the cells, and the Wnt are secreted. And when they are secreted by not exosomal pathway, they will kind of be caught on the Glypican on their way out. And then you have a mechanism that will release the GPI anchor, and they will start diffusing. Is that?
Okay, so I'll there's kind of 2. I'll address the first bit. So I didn't go into it today, but it was work I was involved in my PhD. The lipid also creates a problem in the secreting cells, because how do you get this lipid and stop it embedding in the membrane and being stuck in the EOR or the Golgi? And in the secreting cells, there's a protein called Wnt that basically has a lipid binding pocket. Beautiful crystal structure just came out showing it. And this basically shields the lipid inside the cells and it chaperones the Wnt to the surface. And then nobody knows exactly how you get handover or release from Wintless. And it's probably one of the big questions left in the secretion diffusion field. But at some point you must get Wintless releasing the Wnt and freeing up the lipid and transferring it to DLP. And that could happen at the plasma membrane, or some people believe that you would get internalization of Wintless and the Wnt, and maybe in the hydrophobic endosome, you trigger release and then you get handover to DLP. So that would be one way of getting the handover. And then for the actual diffusion, I don't believe that you get cleavage of the GPI anchor and a release of a soluble glypican. There are proteins that do cleave off GPI anchors. Most of the glypican is still containing the GPI, because if you add these proteins that cleave the GPI anchor, you can see a massive loss and shedding of DLP. So most of it there is containing the GPI anchor. And what I believe happens is that you either get this bucket brigade where the GPI anchored glypican is bound to the Wnt, and just an off rate at a certain frequency occurs and the Wnt diffuses to the next DLP. Or maybe you get cytonemes helping transport from cell to cell. But my preferred model is that there's an on-off rate. So when it's on, it's shielded and happy, and the off rate gives this opportunity to engage the frizzled receptor, or an opportunity for other lipid binding proteins like notum, which is known to cleave off the lipid, access to do its job. So I think it's crucial that there is this access, and I favor an on-off model, but that's just my take.
So just to follow up, there could be like cell protrusion, membrane protrusion would move the Glypican carrying the Wnt a little bit further up, like closer to the receptor, for instance.
Yeah, that's one hypothesis, and there's lots of people who have shown these protrusions play a role. But also, these sugar chains attached to the glypicans are pretty huge, and they probably even bridge cell-to-cell gaps. So while I'm not ruling out cytonemes, I'm staying neutral on that. You could also just get transport along these sugar chains with the lipid briefly unshielded, but the cytonemes could also play a role.
Okay, thank you very much.
Okay, we go to Team In. Please unmute yourself and ask your question.
Hi, is it possible? No, yeah. I'm just wondering, what is the role of the interaction for the secreting cells? Like, because Wnt also goes through a lot of maturation, and then if then, as in your model, you showed that DLP also encounter and capture it inside the cell. Would this play a role for secretion?
So I don't think it plays a role for secretion. In the secreting cells, that function is fulfilled by this protein Wintless, which is a lipid binding pocket. And if you make mutant clones of DLP and you make the secreting cells themselves mutant for DLP, Wnt still gets to the surface of the cell fine. So I don't think it would be involved in transport through the secretory system. That will be fulfilled by Wintless.
Yeah, thank you.
All right. Thank you very much. Don't hang up, everybody, just yet. Firstly, I would like to say thank you very much, Ian. Don't think that we exosomologists are not afraid of the role of exosomes being questioned in some situations. So just keep at it. We're happy to have those things disproved, and Carolina is giving me a thumbs up. With that, I think we should discuss when the next meeting is happening. Oh, did you have any more questions, Aaron?
No more questions. No.
Okay, that's good. So we can show the slides for the next meeting, please, and thank everybody for participating.
Yeah, so we don't actually have any slides, but yeah, just a massive well, thanks to Jan for leading the questions, and for everyone coming today, and obviously a massive thank you to Ian for taking the time to give us a great talk today and engaging with the questions.